In this class, we'll try to learn about endocrine pancreas. The learning objectives of this class would be list the hormones secreted from endocrine pancreas and name the cells secreting them. Name the scientist who got Nobel Prize for describing the amino acids sequence of insulin. Give a brief note on synthesis, regulation of secretion, and metabolism of insulin. Outline the mechanism of regulation of secretion of insulin. Explain the role of glucose in insulin secretion, classify GLUTs and give their functions, draw a schematic diagram of mechanism of insulin secretion, describe the functions of insulin, list the important functions of glucagon, somatostatin, pancreatic polypeptide, appreciate the importance of insulin glucagon ratio, and comprehend the change in insulin glucagon ratio in fasted and fed states. The digestion, storage, and utilization of nutrients are a process that require higher degree of regulation. Pancreas plays a crucial role in controlling these processes. Pancreas has a two parts, the exocrine pancreas, which constitutes about 80% of the pancreatic tissue that controls digestion, and the endocrine pancreas, which constitutes 1 to 2% of the pancreatic tissue that controls storage and utilization of the nutrients. Endocrine pancreas is an important endocrine gland or organ as it secretes insulin, the only anti-diabetogenic and anti-ketogenic hormone in the body. Endocrine pancreas also secretes glucagon that has a metabolic actions opposite to that of insulin. Glucagon increases the blood glucose and promotes ketogenesis. Coming to physiological anatomy of pancreas, islet of pancreas. The endocrine pancreas, which constitutes less than 2% of the total pancreatic tissue, is formed by islets of Langerhans, named after a German medical student, Paul Langerhans, who first described it in a dog pancreas in 1869. Islets are more abundant in the tail than the head of the body of the pancreas. There are about 1 million islets in human pancreas. Islets are oval in shape with a diameter ranging from 50 microns to 300 microns. Each islet contains about 2500 cells. There are four types of cells present in the islets. They're called as beta cells, alpha cells, delta cells, and F cells. Beta cells secretes insulin, alpha cells secretes glucagon, delta cells secretes somatostatin, F cells secrete pancreatic polypeptide. Beta cells constitute around 70 to 80% of the islet cells that secretes insulin. The cells are placed centrally in the islet. The diameter of each cell is around 10 to 12, 10 to 20 micrometers. Packets of insulin are present in the form of secretory granules in the cell cytoplasm that measure about 0.25 micrometers. So this is the diagram showing the the histology of pancreas of islets. You can see the islets of Langerhans. This is the pancreatic acini. So these are alpha cells, these are beta cells, these are delta cells. Each packet of insulin is present in a vesicle and a clear space or halo exists between the wall of the vesicle and the packet. A developed system of microtubules and microfilaments are present in the cytoplasm that facilitates exocytosis of vesicles. Alpha cells constitute around 15 to 20% of the islets and they secrete glucagon. They are present in the periphery of the islet. Delta cells constitute around 10% of the islets that secrete somatostatin, and these cells are typically located toward the periphery between the beta and alpha cells. F cells constitute around 1 to 2% of the islet cells and secrete pancreatic polypeptide. The cells are distributed randomly among the D cells. Islets is highly vascular. They receive about 10% of the total pancreatic blood supply, and through the constitute around 1% of the pancreatic tissue. There is a portal arrangement of the blood vessels of the pancreas which allows hormones secreted from the cell groups to reach the other islet cells. This arrangement is essential for paracrine regulation of hormone secretion from islet cells. The islet cells are neuroectodermal in development. They receive both parasympathetic and sympathetic innervation. Usually, stimulation of beta adrenergic receptor increases insulin secretion and alpha adrenergic receptor decreases it. Increased vagal activity, it is going to increase the insulin secretion. Synthesis of insulin. Like other peptide hormones, insulin is synthesized as pre-proinsulin containing 110 amino acids. The insulin gene belongs to the superfamily of genes that encode synthesis of variety of insulin-like growth factors. Insulin gene is located on the short arm of the chromosome 11. Insulin is the first formed as a pre-proinsulin in the ribosome of rough endoplasmic reticulum. The pre-proinsulin contains four sequential peptides: an N-terminal signal peptide, beta chain of the insulin, a connecting peptide that is C peptide, and A chain of the insulin. So this is a diagram showing uh, structure of the proinsulin from the endoplasmic reticulum. Note that the molecule of containing A and beta chains are connected with a C peptide. Uh, then A and B chains are connected by C peptide. In the structure B, insulin molecule is packaged in a granule of the Golgi apparatus after its synthesis. These granules contains insulin and the polypeptide, the C peptide. These are some of the important factors that regulate insulin secretion. Factors that increase insulin secretion are carbohydrates like glucose, galactose, mannose, glyceraldehyde. Uh, the hyperglycemia is an important stimulator. Amino acids like arginine, lysine, leucine, alanine, ketoacids, free fatty acids, especially long chain free fatty acids, hormones like GIP, gastrin, secretin, glucagon, GLP-1, cholecystokinin, increase vagal activity like acetylcholine, beta adrenergic receptor agonist, potassium and calcium, theophylline. And these are some of the factors that decrease insulin secretion. That is hypoglycemia as occurs in fasting and exercise, somatostatin, leptin, interleukin 1, alpha adrenergic agonist and beta blockers, hypokalemia, beta cell poisons, and inhibitors of microtubules like colchicine. They are some of the factors which are going to decrease the insulin secretion. Mechanism of glucose induced insulin secretion. The glucose enters the beta cells of the pancreas via GLUT2. In the beta cells, glucose is utilized by glycolytic enzymes to pyruvate that enters the TCA cycle to produce ATP. Increased intracellular ATP inhibits ATP sensitive potassium channels, which increases the cytosolic potassium by decreasing potassium efflux. So this is going to depolarize B cells, which in turn opens the calcium channels. Calcium influx increases the cytoplasmic calcium that facilitates the calcium mediated exocytosis of insulin granules. So plasma potassium is a natural regulator of insulin secretion. Hypokalemia decreases insulin secretion. So this is a diagram showing the mechanism of action of insulin. Okay, so insulin is going to bind to its receptor on the cell membrane, okay, and then it is going to bring changes in the tyrosine kinase, uh, and it is going to affect the protein synthesis, fat synthesis, glucose synthesis, and growth and expression, growth and gene expression. So this is how mechanism of insulin secretion from beta cells. Okay, so glucose enters through the GLUT2, and glucose is going to enter into the TCA cycle, which produces the ATPs. ATPs inhibit the potassium efflux, so that leads to depolarization. That depolarization increases the calcium level in the cells, which is going to act on the insulin granules, and insulin granules uh, they exocytosize the insulin. So this is the relation between plasma glucose and the rate of insulin secretion. Um, from the plasma glucose levels 50 to around 300 milligrams. Okay, so as the plasma glucose level increases, the insulin secretion proportionately increases. So this is another mechanism of action of the insulin by different molecular pathways. Okay, so they are going to increase the gene transcription uh, through the different pathways. Okay, and they're also going to control the uh, metabolism of the glucose by mainly uh, AKT pathway. So this is another diagram showing the mechanism of insulin release. Okay, so when glucose enters, it enters into the uh, uh, cycle, and that increases the ATP, that reduces the potassium efflux, that leads to depolarization. That depolarization leads to entry of calcium, and calcium degranulates the uh, insulin granules, so that insulin is released. Insulin is secreted in the fed state, that is the at the time of nutrient abundance. Therefore, insulin is known as hormone of abundance. Insulin ensures optimum utilization of the nutrients. Insulin facilitates the storage of nutrients when nutrients are present in excess uh, of the energy need. It also suppresses the mobilization of endogenous substrates. The stored nutrients are made available at the time of need, like exercise, fasting, etc. Insulin produces its target effect by acting mainly on the liver, adipose tissue, and skeletal muscles. Insulin increases glucose entry into the cell, stimulates its oxidation, and also promotes its storage. At the time, insulin inhibits glucose production. Therefore, the primary function of the insulin is to lower the plasma glucose concentration. It is the only hormone that decreases plasma glucose level. That is, it decreases the basal glucose level and also prevent rise in plasma glucose following feeding. Insulin is the only effective antidiabetogenic hormone. The antidiabetogenic functions of insulin are mediated by its action on liver, adipose tissue, and muscle. So this is the flow chart showing the its action on the carbohydrate mechanism. So in the liver, it increases the glucose entry, converts glucose into glucose 6 phosphate, stimulates glycolysis. It increases glycogen synthesis by inhibiting the glycogenolysis and inhibiting gluconeogenesis. In adipose tissue, it increases the glucose entry, it increases the hexokinase activity, converts glucose to alpha glycerophosphate. It increases the esterification of the fatty acids and stores the fatty acids as a triglyceride, and it increases the storage of glucose. In the skeletal muscle, it increases the glucose entry in the cell, increases the hexokinase activity, increases the oxidation of glucose, and stores it as a glycogen. So that increases the storage and utilization of the glucose, and ultimately it will lead to decrease in the plasma glucose. This diagram showing the action of insulin on the adipose tissue. Okay, so in the glucose entry in the adipose tissue is increased, uh, it gets converted into alpha glycerophosphates. Okay, so it is going to uh, uh, decrease the lipolysis. It is going to act on the hormone sensitive lipase to decrease the lipolysis, and it helps in the storage of the fat. This is the action of, or rather, inhibition of ketoacid formation by the insulin. Okay, so it is going to stimulate the conversion of acetyl CoA into malonyl CoA. Okay, so that uh, rather inhibits the reduced formation of the ketoacids from free fatty acids. So the protein metabolism, it is going to, increase the uptake of amino acids from liver and skeletal muscle tissue. Uh, in there, it is going to increase the protein synthesis by increasing the NRA activity, and it is also going to decrease the lysosomal activity. So there will be decrease in the proteolysis. So this chart uh, describes the insulin action on the liver. It is going to decrease glucose output, that is glycogenesis, decrease glycogenolysis, and decrease gluconeogenesis. It is going to increase protein synthesis, increases the lipogenesis, decrease ketogenesis. In the skeletal muscle, it increases the glucose entry, it increases the glycogen synthesis, it increases amino acids uptake, decreases proteolysis, decreases gluconeogenic amino acid release, it increases the potassium uptake. Whereas in adipose tissue, it increases glucose entry, it increases GP synthesis, it increases the hormone sensitive lipase, inhibits lipolysis, promotes lipogenesis. So overall, it is going to decrease plasma glucose levels, decrease plasma amino acids, decrease plasma potassium, increase protein synthesis, decrease protein degradation, decrease lipolysis, increase lipogenesis. Insulin deficiency, that is diabetes mellitus. Deficiency of insulin results in diabetes mellitus. Diabetes mellitus is the most common endocrine disorder in both developing and developed worlds. Presently, the incidence of diabetes mellitus in India is increasing like an epidemic. Every one in three uh, in urban and every one in five in rural population are found to have a latent or frank diabetes mellitus. Insulin excess occurs in insulin secreting tumors of pancreas like insulinoma. This causes hypoglycemia. Chronic hypoglycemia causes incoordination of the movement and slurring of the speech. So this is usually misdiagnosed as a drunkenness. It is typically most common in morning as towards the early morning blood glucose concentration decreases and hepatic glycogen store is depleted. Hypoglycemia also occurs in excess administration of insulin in the treatment of diabetes. But in this condition, hypoglycemia is acute and manifest in the form of sweating, palpitation, anxiety, and other autonomic functions. Glucagon. Glucagon has the opposite metabolic function that of insulin. It is pro-diabetogenic and ketogenic hormone. Glucagon is secreted from alpha cells of pancreas, which constitute around 20 to 25% of the islet cell mass. Glucagon is also secreted in the intestine, that is as a enteroglucagon, as a local hormone and found in some parts of the brain as a neurotransmitter. It is a polypeptide hormone containing 29 amino acids. The molecular weight is around 3500. Like other peptide hormones, it is synthesized as a Pre-proglucagon that has 179 amino acids. Pre-proglucagon is found in alpha cells of the pancreas, L cells of the terminal part of the intestine, and in the brain. Pre-proglucagon molecule is the alpha cell contain glucagon and major proglucagon fragment and glycine interrelated polypeptide, whereas pre-proglucagon molecule in L cell of the intestine contains glycatin, glucagon like polypeptides GLP-1 and 2, oxytomodulin, and MPGF. Accordingly, pre-proglucagon molecule forms different hormones in alpha cells and L cells. In alpha cells, it forms proglucagon that finally forms glucagon. Glucagon secretion is regulated by the following factors. Factors that increase secretion are amino acids, especially gluconeogenic amino acids, cortisol, CCK, gastrin, stress, exercise, infection, beta receptor antagonist, and theophylline. Factors that decrease glucagon secretion are glucose, somatostatin, insulin, secretin, ketone bodies, free fatty acids, and alpha receptor agonists. Glucagon is mainly degraded in the liver. It circulates freely in the blood. Its half-life is around 5 to 10 minutes. Glucagon acts by increasing cyclic AMP in the cell. It also increases IP3 in the cells. Physiological actions of glucagon are almost opposite to that of insulin. It facilitates mobilization of nutrients by decreasing their storage. It specially facilitates hepatic glucose output and ketogenesis. On carbohydrate, it stimulates glycogenolysis. It causes immediate and profound glycogenolysis in the liver by activating glycogen phosphorylase. This causes release of glucose 6 phosphate. It also stimulates gluconeogenesis by activating gluconeogenic enzymes, especially phosphoenol pyruvate carboxykinase. Glucagon inhibits glycogen synthase. Therefore, conversion of glucose to glycogen is inhibited. Or the fat metabolism, it promotes lipolysis and ketogenesis in the liver. In hepatic cells, glucagon facilitates lipolysis. Thus, it increases the release of free fatty acids from the liver. However, the fat store of liver not more unlike adipose tissue. Therefore, the degree of lipolysis is moderate. Glucagon increases ketone body formation in the liver cells by decreasing the level of malonyl CoA. Increased, increased free fatty acids also promotes ketogenesis. Glucagon facilitates the entry of free fatty acids into the mitochondria for the beta oxidation and production of ketone bodies. Insulin glucagon ratio. Insulin is a glucopenic, glycopenic, anti-diabeto, anti-gluconeogenic, anti-lipolytic, and anti-ketotic. Okay. So it acts as an anti-diabetogenic and anti-ketogenic. Therefore, insulin is the hormone of energy storage, whereas glucagon is a pro-diabetogenic and pro-ketogenic. Therefore, glucagon is the hormone of energy release. Because of their opposing effects, uh, a balance should be maintained between the secretion of insulin and glucagon for maintaining normal metabolic functions. Therefore, insulin glucagon molar ratio, that is Ig ratio, in the plasma is more important than their individual concentration. Normally, Ig ratio following a balanced diet is approximately 3. Following overnight fasting, it decreases to 1, and after prolonged fasting, the ratio may be as low as zero point 0.4. Following glucose infusion, the ratio may rise up to 30. During starvation, low Ig ratio favors glycogen breakdown and gluconeogenesis that increases the nutrient supply. During high fat state, the ratio is high, favoring deposition of nutrients in the form of glycogen, protein, and fat. During neonatal period, immediately after birth, a low Ig ratio is critical for the survival of the neonate. In the few hours to days, the abrupt cessation of maternal fuel supply, during which the neonate does not receive the required amount of fuel from its own GI tract, a low Ig ratio maintains the internal supply of nutrients. In diabetes, inappropriate Ig ratio influence the metabolic status. Glucagon excess is produced by tumor of alpha cells of pancreas. So this is called as glucagonoma. It causes hyperglycemia, ketosis, and other metabolic derangements that mimic diabetes mellitus. In fact, insulin deficiency in diabetes is associated with glucagon excess. Glucagon deficiency occurs rarely. So this is in brief about insulin and glucagon. Thank you.